Hello friends, this is the final lesson of the course Biotechnology, its tools and techniques, and this lesson is about recombinant DNA technology. In this lesson we are going to learn about this topic in detail. This is about me, and okay, first of all, what is it? What is recombinant DNA technology? Recombinant DNA technology is also known as genetic engineering or cloning. It is the set of techniques that enables DNA from different sources to be identified, isolated, and recombined so that new characteristics can be introduced into an organism. One of the main techniques that is used in recombinant DNA technology is to produce a recombinant DNA molecule. And by recombinant DNA molecule, we mean a molecule which is having DNA from two different sources or two different organisms. Generally, one organism is a vector, act as vector, and into which the desired fragment has been inserted. This desired fragment is inserted into the vector with the purpose to produce an unlimited number of copies of this segment. And now the question is, what is a vector? Vector is a molecule which can autonomously replicate itself on its own when it is inserted into suitable host. So, a recombinant DNA molecule is a chimeric DNA molecule having DNA from two different sources. One which is able to replicate itself, having all the regulatory sequences which are needed for its replication in the host molecule, and other source of DNA is the desired fragment whose multiple copies we want to produce, or whose for it is the sequence whose different clones we want to produce. This technique was given by Stanley Norman Cohen and Herbert Boyer in 1973. And now the question is, why we are doing all this all these things, and what is the need of producing a recombinant DNA molecule? Generally, there are two major objectives to produce recombinant DNA molecule. One is the large scale protein production, and second is the gene expression. When we mean to say that large scale protein production, the vector, the desired fragment which is inserted into the vector is introduced into the host, and within the host the desired fragment shows its expression, and the expression of the desired fragments is controlled by the regulatory sequences which are present in the vector molecule. So by this way the gene is expressed, and most of the times this gene is coding for a particular protein, and since the protein is produced within the host and it can be obtained from the host organism. Second is the gene expression. When we want to see the phenotype, or we want to know the function of a particular gene, it is inserted into the vector and it is introduced into the host. By using the regulatory sequences of the vector molecule, this gene expresses itself, and the particular phenotype of the gene can be scored and can be identified by this method. Now coming to next slide. How? Multiple copies of the gene can be obtained by various techniques. Some of the common technique is gene cloning, PCR, and gene synthesis. Gene cloning or DNA cloning is the production of large number of identical DNA molecules from single ancestral DNA molecule. The essential characteristic of DNA cloning is that desired DNA fragments must be selectively amplified, resulting in large increase in copy number of selected DNA fragment. This involves multiple rounds of DNA replication. Now, DNA cloning approaches can be of two types: cell-based or cell-free DNA cloning. When we say gene cloning, it is the cell-based DNA cloning, where we produce a recombinant DNA molecule having the vector sequence and the desired sequence, and it is introduced into the suitable host, most of the time the E. coli. And when other technique of DNA cloning is cell-free DNA cloning, and the most common technique which is used for cell-free DNA cloning is the PCR, or polymerase chain reaction. I have talked about this PCR in detail in my previous slide in lesson 1, so if you have any query you can take that lesson and you can understand about this technique in detail there. Now, how all this is done? By cutting the DNA into suitable fragments by restriction enzyme, and it is then joined to form chimeric gene having regulatory sequence from one organism and gene sequence from other. As I have told earlier, the DNA fragment or the desired DNA fragment is cutted from the source DNA, from the whole genomic DNA, so that we can selectively introduce this particular DNA fragment into the vector molecule, and after insertion into the vector, it is introduced into the host, and after introduction into the host, this particular fragment is selectively amplified irrespective of the source genomic DNA. So we can produce multiple copies of the single desired fragment, and this can be done by this technique which is known as recombinant DNA technique. Now steps is the production and isolation of DNA fragments to be cloned. That is when we have a whole genomic DNA of a particular organism, and we want to clone a gene sequence which is coding for a protein which is have some commercial application. So from the whole genomic DNA, this particular gene is isolated through restriction enzyme digestion or through some other technique, and this is isolated, and this fragment can be cloned further. Now this particular fragment is inserted into the suitable vector. Vector, as I have told earlier, is a molecule which is having autonomously replicating sites within itself, and with the help of these sites, it can on its own produce its copies within the host molecule. Next, after the insertion into the suitable vector, this recombinant molecule is introduced into the suitable host, which is most of the times E. coli bacteria. It is introduced into the E. coli bacteria, and now from the culture after introduction through there are various methods of introduction of recombinant molecule within the host, such as electroporation, chemical transfer of this CaCl2 base transfer into the competent cells of the host molecule, and after insertion into the host molecule, that from the whole culture of the host molecule, the transformed cells are selected. These transformed cells can be selected on the basis of the selectable marker which is present within the vector molecule. The vector molecule along with the ARS site also has some selectable markers. Most of the times it is an antibiotic resistance gene, and this whole culture is plated on a medium plate which is having antibiotic, and all the cells which are non-transformed, they remain susceptible to the antibiotic and they dies, while the transformed cells, because of having introduced vector molecule, recombinant vector molecule, they grows in presence of antibiotic, because this vector is having some antibiotic resistance gene within it. Now in this way we can select the transformed host cells, and from these transformed host cells we pick the colonies of the transformed host cells, and we can again suspend them into the liquid broth medium so that we can prepare the whole cell culture of the transformed cells, and from this transformed cells we can again multiply this transform, we can again isolate the desired fragment through plasmid isolation, and we can, if we are doing it for protein production, we can even isolate the protein for which the gene of the desired fragment was coded. Now next slide is about what are the different tools used in it. As it's a very complex technique, there are various different tools which are used, and these are the different molecules which are acting as tools here. First is restriction endonucleases. As I have told in my previous slides, that restriction endonucleases are the molecules or enzymes which act as a scissor, molecular scissor. They cuts the DNA, genomic DNA or whole DNA segment into smaller fragments, so that these fragments can be inserted or into the vector molecule. Now for insertion or for joining of these desired fragments with the vector molecule, we need another enzyme, which is DNA ligase, which is responsible for joining the DNA fragments with the vector molecule. Next is reverse transcriptase. Reverse transcriptase is an enzyme which is used for the production of mRNA from the which is used for the production of cDNA from the mRNA molecules. And just like this, there are many other enzymes which are used for the purpose, such as alkaline phosphatase, which is polynucleotide kinase, and S1 nuclease. S1 nuclease is again an endonuclease which is purified from Aspergillus oryzae, and this enzyme degrades RNA or single-stranded DNA, but it does not degrade RNA DNA hybrid or DNA duplex. So in case of some complex kind of gene cloning, we use S1 nuclease as well. And like alkaline phosphatase and polynucleotide kinase, they have contradictory function. Like alkaline phosphatase is used for the addition for the removal of phosphate from the DNA fragments, while polynucleotide kinase is used for the addition of phosphate group. Thank you. I hope I'm clear about what is RDT, that is recombinant DNA technology, and what are the tools which are used for the purpose. And if we want to talk about the what are different applications of this technique, there can be different applications, like the ability to manipulate DNA sequence for the molecular diagnosis of disease, for the production of vaccines, for gene therapy, and for cloning a particular DNA segment from a trait, and for the plant variety production, or animal breeding as well. So recombinant DNA technology is having vast applications, and it is a very complex and sophisticated lab technique. It needs very skilled laboratory workers who can work for this for doing all these techniques. I hope you all liked my video, and if you have any query, you can comment and you can let me know about that. Thank you.